Selamat Hari Raya, Maaf Zahir Batin to all Muslim students. Okay. And I think masih lagi beraya, semua orang masih lagi beraya. And hopefully semua orang bersemangat untuk kelas hari ini. Okay, I think this is your first class, of course. First class after Raya. Yeah. Okay. Yeah, I'm going to share slides. Okay. Okay, so last week pula dah last week Raya. Okay, last 2 weeks. Okay, last 2 weeks we end up with the topic for photosynthesis, right? So we have go through on the steps, okay, step by step on how the protein, on how one peptide is produced, okay, is synthesized, okay. It involves how many steps for photosynthesis? Dah lupa dah. Makan kuih raya banyak sangat. Okay. So how many steps are they for photosynthesis? Okay, it starts with the activation, right? Activation, and then the next one, what do we have? Activation, initiate, yeah, initiations, all right. And then we have the elongation, and then the last part is the terminations and release. Okay, termination and release. And we have another one, which is the process of folding, right? So to make sure that the protein is functional, right? So that is the functions for folding. And we're going to look at today, not in details, just to see relationship or some more continuations of the photosynthesis to folding, right? So if you look at here at the cover for the slides today, so we have the photosynthesis process starting from DNA, and then we have the translations, right? The translations process happen, and then how the proteins that which is synthesized becomes first thing is that the protein is unfolded, but then it should be folded. It should be folded for the protein to be activated, and in this case somehow the protein can go through a folding process, through many steps, okay, many steps and many options, like we have previously we have discussed, which is involve modifications on the N-terminal parts, C-terminal parts. Yeah, so please check them. Okay, check them again. What types of modifications that we have discussed on the previous weeks, right? And then, and we also have the the collaborations, with the chaperones, okay, in order to fold the proteins, okay, in order to fold the proteins, and we get the functional complex, which is the functional proteins. All right, and if let's say the folded unfolded proteins is misfolded. Okay, misfolded. It goes through the process of folding, but then it is misfolded. Then we have to use the chaperone. Okay, we have to use the chaperones so that it can be folded. It can be folded and become the functional complex. All right. And in another case, if let's say the misfolded protein is not successfully successfully refolding, okay, refolding by using the chaperones or other types of methods, then the misfolded protein might be end up with aggregated protein, which is not active, which is not functional, right? Not fully functional. And okay, so, and we know that protein synthesis is we have discussed on the how the protein is synthesized to form a long polypeptides, right? And at the end of it, we have a long polypeptides, which is the products, okay, the products from decoding process of the DNA or genetic, okay, or genetic in the nucleus. All right. And all right, so we have discussed on the protein synthesis of the step, okay, this step by step on how the protein is synthesized. So in the nucleus, we have the transcription process takes place, which is the gene or DNA is transcripted into this long RNA, okay, long sequence RNA here. And then it will go through this nucleus pore as the mRNA, okay, as mRNA or messenger RNA, which brings all the information that's already decoded from the DNA sequence. All right. So, and from here, this mRNA will go through the process of translations. Okay, translations, and here we need many factors. Okay, many factors, for example, we have discussed before the elongation factors, and yeah, so many factors that will help to go through this protein synthesis. Okay, so, and we have here is large and small subunits of ribosomes, which is a very important ribosomes. Okay, very important ribosomes to actually guide the process of elongations, okay, elongations and translocations of the protein synthesis. All right. So here we have what we have discussed before. So we have four steps, okay, four main steps in the process of protein synthesis, which is activations, initiations, elongation, and termination and release. And we have the another one, which is the last steps. It's actually not included in photosynthesis. The last step, which is the modification and so on, that is another another big step lah, another category. Okay, another category of protein, on how protein is modified. Yeah. So we have here protein synthesis. So we have the 4 steps, but this is the 4 steps that involve in protein synthesis: activation, initiation, elongation, termination, and release. Yeah. Okay. And so these figures here we can simplify. Okay, we can simplify on how this DNA and then transcripted into RNA, and then we have this sequence of the mRNA to be translated, and we get the long polypeptides of protein. Yeah. Okay. And so just to refresh again on what we have discussed on the translation process that takes place by having these 3 main steps: initiations, elongation, and terminations. Okay, so last time we have discussed on how we need the initiator of methionine tRNA. So during the initiations, we need the tRNA which contain or which carry the methionine, which bring the signal of to start protein synthesis. So that is the methionine which brings methionine tRNA. And we also need the mRNA bind to the small ribosomes subunits to initiate the protein synthesis. And so we have the initiations where we have the small subunits bind at first to the mRNA, and then we have the large subunit comes in. So we have the initiations complex, remember? Okay, initiation complex. And then during the elongations, we are going to have the next amino acids, which attach to the tRNA, okay, to the which will occupy the A site. Okay, if you still remember, we have the 3 sites at the ribosomes. Okay, the first one is A site, P site, and E site, right? Okay, so please refer again to the notes. Okay, and here the elongation will start. Okay, and then so the thing is that if you remember last time we have discussed on how the ribosomes moved, okay, during the translocation process. Okay, so here, what can you understand? Which part is actually moving during the translocation? Whether it's ribosomes moved or the mRNA actually moved? Which one? Okay, during the translocation, which one do you think moved? Whether the ribosomes or the mRNA?
Ribosome.
Alright. Okay, ribosomes. Ada yang cakap mRNA. Apa sebenarnya yang bergerak during the translocation? Sometimes, sometimes this part is quite confusing. Okay, sometimes this part is quite confusing, but I need you to be clear on this. Okay, if you tengok video ke protein synthesis, might be helpful for you to understand the process. Yeah. Okay, yang bergerak adalah ribosomes, yang bergerak, yang the one that move during the translocation is the ribosomes. mRNA dia tak bergerak. Okay, mRNA ni dia static kat situ je, one strand, and then the one that move is the ribosome. Okay, ribosome akan pergi ke next codon and next codon, and so once it move, the new codon, the next one, akan berada pada A site. akan berada pada A site, dan begitulah seterusnya for the previous previous tRNA, or akan berada pada akan occupy the E site. So sebab ribosom tu bergerak. Okay, so jadi akan berlakunya perubahan siapa yang which amino acids akan occupy the site dekat ribosom. Okay, so that is the thing. So you can actually watch banyak sebenarnya video. Okay, so there are so many videos that will be helpful for you to understand the process clearly. Okay, dekat YouTube. So so I just stress on this one because sometimes students, sometimes some students, they even not aware of this kind of movement, whether the ribosomes or mRNA yang sebenarnya bergerak. Alright. And okay, so at the last part, which is terminations, we know that we need the termination codon, right? So termination codon, I know that you we have discussed on this one. So we have 3 types of stop codon. So whenever they found this stop codon anywhere in the mRNA, then it will give the signal to stop the synthesis of the protein, right? Okay. So when we talk about the protein, the polypeptides, the polypeptides. So what are the proteins made of? Okay, what are the proteins made of? So yeah, of course, because it consists of the amino acids, right? Consists of the amino acids, because we can say that the proteins actually the building blocks of amino acids. Banyak amino acids, and they bind together during the process that we have discussed before, during the translation and elongation semua tu, until at the end of terminations, then we have the long polypeptides. So we can say that the protein itself is the building blocks of the amino acids. And what are actually contained in one single amino acid? Okay. Yang ni sebenarnya dah masuk exam hari tu kan, because this is very basic sebenarnya. And tak apalah, we discuss again. So this is the answer lah for your exam hari tu. So in one amino acid we have the amino group, kan? Amino group, we have the carboxyl group, we have the hydrogen, and then we have the R, which is the side chains, which determine the characteristic or the properties of each amino acid, whether they are actually the hydrophobic, the hydrophilic, whether they are polar, non-polar. So that is the role of the side chain. But the side, the other side, okay, the carboxyl group, amino group, and the H here should be the same in all amino acids. Just the thing that it just have different configurations over here, the side chains. Yeah, all right. And how does this protein, this amino acid, is combined together, from one amino acid to the next one, which happen in the elongation process in the protein synthesis. So so here's the thing, right? So previously, this is also in your exam, and macam mana the amino acids attached to the next one. So it has to be the carboxyl group, carboxyl group attached to the amino group to the next amino acid, right? So here we have the CO. So initially, if it's just one amino acid, we have COOH, right? CO and OH here, right? Ini semua jawapan kepada test 1 tempoh hari. Ada yang betul, which is good, meaning that dia tahulah, this is very basic for biochemistry students. So she or he prepared themselves for this kind of questions. Okay, so we have Tak nampak sangat kan, gelap sikit ni sini. So it should macam ni. So COOH. So when this group COOH attached to the next amino acids, so the next amino acids, the group of the N terminal of the next amino acid should be attached to the carboxyl or carbonyl carbon of the carboxyl group. Macam ni. So we have the N terminal of the next amino acid over here, NH. So it attached to the carboxyl carbon of the carboxyl group. Okay, right. So akan muncul lah peptide bond ya, peptide bond so between the N and C. Okay, so this is peptide bond. Okay, and when we have a long sequence of amino acids, when they attach together to the next amino acid, to the next one, and to the next one, which perhaps the R chain, the R side chains here is going to be different. Okay, so they have a different properties and they attach together. Okay, side by side like this. We can say this is a primary, primary structure of the proteins. And since they have a different properties for each of the side chains, okay, so they tend to make an interaction between one amino acid to another one in a different dimension, okay, in different dimensions. So when this happens, the protein is going to be, okay, dia akan folding, which is what we can say this protein folding is going to help the protein to be activated. And for such arrangement, for such three-dimensional proteins, they are going to have a proper site for the substrate to attach to, and they are going to do any types of process. Protein, it can be enzymes, it can be a hormone, it can be anything. Yeah, the protein, right? Okay, so these are the 20 amino acids that we have discussed. Okay. All right, so here is not actually 20, it's 21 because including the selenocysteine. Okay, selenocysteine, it has the combinations of the selenocysteine here instead of cysteine, just cysteine, but it has selenocysteine. So 20 plus 1 lah, according to this chart. But basically, selenocysteine is quite not really common. Okay, not really common, but it's stated here. Anyway, so we have 20 amino acids. In 20 amino acids, we have different categories of them, which is some of it, they are actually negatively charged amino acids, positively charged amino acids. Yeah. And then we have also amino acid with a polar uncharged side chain, which is polar amino acids. And we have the hydrophobic side chains, okay, many of it, okay, with a hydrophobic side chain. So this also very important for you to remember, okay, for you to remember all these 20 amino acids, yeah. And it's very easy to memorize all this. You can find actually many tricks, okay, many tips to memorize this, to memorize all the structure and the properties of the amino acids. Yeah, you can find out banyak cara, many types, many different types, many different tips as well to actually help you to memorize all these 20 amino acids. It's just that you have to find the one that's sesuai lah, okay, sesuai dengan you punya cara, okay. Alright. And okay, so during this the elongation process that we have discussed on the photosynthesis. Um, so there is one part during the elongation process where there is a reactions that happen. This is a very important reactions mechanism of peptide bond formations. Perhaps we haven't discussed in detail on how the peptide bond formation happen, right? It's just stated that okay, so the end terminal part of the um, um, second amino acid just attached to the carbon of the carboxyl group of the first amino acids. But then, how does it happen? How the mechanism takes place? Okay, how does the mechanisms happen? Um, so here actually we have the N-terminal part of the second amino acid, right? So the N-terminal, the amino group here is very nucleophilic, nucleophilic, yeah, because this is highly negatively charged. The N, the nitrogen, is highly negatively charged. Yeah, so it's a very nucleophilic. So it tends to make interactions to the highly positively charged of the carbon of the carboxyl group. So what happens is that they will the end will attack the carboxyl group, okay, of the first amino acids, all right, and then the hydroxyl group OH, okay, OH of the carboxyl group is going to be released from here, okay, and uh, then we have another proton here, which is extracted or will be pulled from this hydrogen from this amino group. So meaning that we have okay. So since we have COOH, right? So what happened is that the end over here is going to attack the carboxyl group, and it will force the CO to clash, okay, to to just push it over, and we are going to have the OH. And since OH is a very negatively charged, and it's going to pull the one of the hydrogen from the amino group. So to here, so that's why the peptide formation is going to release the water, all right, H2O, because it comes from the combination of OH from the carboxyl groups with the proton from this amino group of the second amino acids. Then we have the H2O. So that's why during the peptide formations, they will release water. Yeah? So this is how it happens. So we have discussed on the mechanism of peptide bond formations. All right. Okay. So if we refer to what we have discussed before in the process synthesis during the elongation steps, which is the one step of the translation cycle. So the first peptide formation is over here. When the first amino acids attach to the second one. Okay, so they have something like this. Yeah, so this is the first peptide formations, and yeah, it happens exactly like this as we have discussed on the nucleophilic attacks, the electrophilic of the carboxyl carbon. Okay. Yeah. So when we have the primary, when we have the primary sequence or primary structure of the proteins, which is a long polypeptide like this, and because since the R side chain of the amino acids, they have a different properties, and we know that it tends to make an interactions to the another amino acids around surrounding, right? And it depends also in which environments that the protein is the protein is mixed in, okay, whether in water or in lipid. So it depends on what the environment of the amino acids. So they are going to actually behave differently. Okay, and so these amino acids, the long sequence like this, we call this primary protein structure. So it's a sequence of a chain of amino acids. Then if let's say the primary protein structure somehow make an interactions, okay, perhaps because of the interaction with another amino acids, or perhaps with the environment around it, okay, then it can become this alpha helix and also beta sheet, okay, so they can have a different configurations. And after that, with more interactions that they have, they tend to finally what we call this as the tertiary protein structure or three-dimensional folding pattern of a protein. Yeah, because of the side chain interactions from from one amino acid over here to here, or from one amino acid from here to with this one. Yeah, in a primary protein structure, we may say that, oh, this is impossible that the amino acids that located in here is going to interact with the protein over here, right? But that's not the thing, okay? They tend to um, to fold, okay, because of the sections that they have. All right. So finally, the tertiary protein structure, T1 tertiary protein structure, they are going to have a protein-protein interaction with another peptides, polypeptides. So when this happens, we call these conditions as quaternary protein structure, where proteins consist more than one chain. Okay, they have more than one chain, meaning that they have more than one subunit. Okay, it has totally one subunit over here, and we have another second subunit, which they need the combination of these subunits to be functional, right? So this is what we call as quaternary protein structure. Okay. So this is one of the example of folded protein or functional proteins, which is the protein bacteriorhodopsin. And for information, so this bacteriorhodopsin is a type of membrane protein, in bacteria that will help for the proton pumping, proton pump from the inner side to the outside of the membrane, or from the outside to the inside. So it depends on the flexibility of the membrane proteins, or of the mechanisms or metabolisms of the bacteria itself. And as you may see, we have so many alpha helix. Okay, 1, 2, 3, 4, 5, 6. So it consists of 6 alpha helix, and it also has the beta sheet in red like this. Okay, and we have even the coil. So this is the shape of 3D structure of the functional protein for the protein bacteriorhodopsins. Yeah. Okay. Okay. What happens? Why protein is folded? Okay, is folded because we know that protein folding is very important for so that the protein becomes functional, right? Okay, so this is the unfolded proteins. Unfolded meaning that perhaps this is the situations that we have if if the photosynthesis is completed, yeah. So, but then these types of proteins need to be folded before they are considered as the functional proteins. Okay, and for that, for the protein to be activated, or if they need to be folded, they are going to need several forms of molecular interactions. So this one again it depends on the R chain, R chain of the amino acids, and of course the interactions, which includes the thermodynamic stability of the complex itself. Okay, and another one is the hydrophobic interactions and the sulfide bonds. that forms in the proteins. So so meaning that here we just not consider the R chain itself and the environment of the protein, the surrounding the proteins, but we also need to consider the thermodynamic stability of the complex. Yeah, thermodynamic stability of the complex and the hydrophobic interactions. This is very important because if let's say the protein is less hydrophobic, is hydrophobic, but then we put the protein into the water, so we might see the protein is going to be degraded or is going to be um, uh, pretty unfold because of because the protein is not stable in the water because they have this kind of very hydrophobic uh, pretty um, amino acids, yeah, inside the protein sequence. Okay. Okay. So the protein folding is actually will determine whether the protein is stable or not. Yeah, will stable or not. Because the folded proteins basically is actually stabilized by thousands um, of non-covalent bonds, yeah, between the amino acids. So this is what makes the protein stable. Once they are folded, that is actually stabilize the proteins since they have these non-covalent bonds between one amino acid to the next one or uh, to other amino acids in its range. And okay, so so over here we have the hydrophobic group. Okay, so we have one strands of the proteins or polypeptides. Saying that if let's say for example we have the hydrophobic group, okay, over here, and we have the and if let's say these proteins, the hydrophobic group is put into the water, okay, so we have water molecules in red and blue, which is represents the H2O, right? So whenever we put these hydrophobic proteins, highly hydrophobic proteins over or into the water, so you can see that the part of the protein or the amino acid is going to be inside. It is going to um, go away from the water. Dia tak nak jumpa dengan air. Okay, the hydrophobic part, because you know we have the part of apa ni if you I think you're quite familiar with the membrane protein, right? Membrane proteins where if we because in membrane protein we have so many lipid where we have the sites that love water and the sites that hate water. So whenever you put the membrane protein or inside of water, so it's going to have a different configurations. So the same thing over here, if we have the proteins with the hydrophobic group like this in in different environments, which is less water, so it's going to have this kind of configurations. But then if we put this protein into this let's say a solutions with the solution of water, so it's going to have a different configurations where the hydrophobic part of the amino acid is going to hide from the water, so you have is going to have a different configurations. So this means that the amino acids or the protein they are going to have a different characteristic. A different characteristic. It depends on the environment itself. So meaning that protein stability normally we're going to refer to physical or thermodynamic stability. So thermodynamic stability is including how the environment in terms of the solutions or temperature, okay, the pH, all those we call it as physical stability that will or physical parameters that will affect the physical stability of the proteins or thermodynamics of the protein. Okay. Okay, so how are we going to increase the protein stability somehow? So there are ways to increase the protein stability. So normally protein or stability of the proteins depends on the extent of the disulfide bond. Okay, so the more or the number of the disulfide bond in a certain proteins, they are going to be more stable, and they are going to um, uh, they are more stable, meaning that they can be um, they are going to have a longer half-life in a higher temperature. So that make it, because of the disulfide bond, makes these proteins highly thermostable. And another thing is that the presence of certain amino acid and the N-terminus part, for example, we take for example the N-terminus of the beta-galactosidase. So let's say the amino acids that contain in the N-terminus, we have the methionine, serine, or alanine. They're going to have a longer half-life. And if let's say they have more arginine in the N-terminus, then they are going to have a shorter half-life. So this is for example of beta-galactosidase. Okay, take for example these enzymes, but different enzyme or different proteins, of course, they are going to have a different content of whatever amino acids that they have in the N-terminus. So it depends. But the principle is that if they have this kind of amino acids, okay, the N-terminus and at the N-terminal, then they are going to have whether they are going to have a longer or shorter half-life, right? And the next one is the internal path sequence. So I think we have discussed actually on this one on the earlier topics, on the earlier weeks previously, on how the internal path sequence and also the part on the N-terminus actually also influence the protein stability. Yeah. So we have the proline, glutamic acid, serine, and threonine. Okay. So this is PAS. Okay. And if we have uh, this PAS region, okay, so make a protein more susceptible to proteolytic degradations. Okay. And the stability will be enhanced by PAS region, could be altered by genetic manipulations. Okay. All right. So this is the energy state of protein folding, meaning that if a certain protein is in the unfolded state, they are going to have higher energy, which is the higher entropy. Okay. And okay, by the way, this is what we call as the protein folding funnel. Okay, protein folding funnel. So the higher the energy, meaning that the protein is actually not stable. Okay. So this is actually the same funnels, man. Same concept. Yeah. So we have the free energy here, free energy here. Okay. So let's just take a look on the left side first. So if we have a certain protein that is unfolded, okay, unfolded, meaning that this protein is actually having the very high energy, okay, high entropy, and if this unfolded protein goes down over here, perhaps because of chaperone helps the protein to fold, then the energy becomes lower. Okay, perhaps now it's become molten global state, then the energy is become over here, become lower. And if the proteins totally folded in an active state or inactive state, then it's going to have a totally lowest energy. Yeah, so that is the concept of protein, fold protein, and this is actually the same thing. Okay, the same thing. It's just that to show the in details on each of the steps or stage that involved during the, starting from the what happened in the higher energy. So perhaps it's actually unfolded like this, and as it's folding in the folding intermediate state one, so the energy becomes lower to here, and for the intermediate state two, then the energy is going to be lower to this one, and yeah, until it has the native structure, and definitely the energy is going to be the lowest, right? Um, so the percentage of release of protein in native conformation lah. So the percentage of the residues in protein conformations. Okay, so it's going to totally in the native structure. Okay, so protein stability is very important for many reasons. Okay, why we want the protein to be stable, to be of course the first step, it need to be fold, and then it need to be stable. So enzymes or proteins is very important for many applications. You need to, especially if the protein is the enzymes, we need the enzyme for biocatalysis. Okay, biocatalysis, or to catalyze the reactions to make it faster. So it's going to be very expensive applications. Then, so let's say if we use a certain chemical, let's say for the certain process, let's say for making drugs, yeah. So the chemical is very expensive, but then if we replace such process with the biocatalyst, which is the enzyme, which is much cheaper, and let's say this enzyme is much stable, much cleaner, then it's going to be a very good way, okay, very highly benefits to the economy as well. And the proteins, it can be used for household items, okay, biofuels, to generate biofuels, pharmaceutical, animal feeds, beverages, and of course in R&D diagnostics. So there are actually many important applications of proteins, functional stable proteins in different fields. Okay. Okay. So this is the final shape of the protein. Actually, I think this slide should be before the application. Actually, sorry for that. So just another addition to what we have discussed on the protein folding. So the protein folding, it can be aided, okay, by chaperone, okay, what we call it as chaperone protein. So if we have such a protein which is unfolded, sometimes the unfolded protein, it cannot be fixed, okay, perhaps because of oxidative damage, and it will totally damage, and it will become aggregated. But in another side, in another way, the unfolded protein, it can become a native state because of first thing, because of the modifications of terminus or C-terminus or another parts of the protein itself. Okay, so we have discussed on that one previously. And another one is an unfolded protein. It can become a native protein or an active protein because of the chaperone. Okay, presence of the chaperone. So the chaperone, it can help the protein to be activated or folded. So this is chaperone protein. So this is the structure of chaperone protein. So what happens is that the protein or unfolded protein, or it will enter like a chamber. So we have, for example, here the GRU-EL. So GRU-EL is a type of chaperone, protein chaperone, and they need the presence of the dia punya pair lah, dia punya kawan dia, GRU-ES. So they will work together to process this unfolded protein initially, and during the process they need the ATP. Okay, and then when they finish the process, then the ATP, of course, it will be consumed during these times, and we have the ADP, and finally we have the properly folded protein. So this is the function of the chaperone proteins. Yeah? And this is a little bit in details, I guess. This is the figures from the outside, yeah, but then this is the figure from the inside, from what happened inside the chamber. So you can see so how this happened. We have the grow EL, okay, grow EL, and then grow ES comes in to cover the grow EL, and we have the unfolded protein and ATP, and they are going to process the unfolded protein inside the chamber, and then we have the folded protein as the products from the GoEL and GoES. Right. So that is the one of the example of the chaperone protein. And actually we have a lot of chaperone proteins, not just GoEL and GoEL, GoEL and GoES. We have so many DNAK, DNAs. I can't tell you because there are so many long list. There is actually a long list of chaperone proteins that we can use to fold the proteins, but it depends on the kesesuaian jugalah. Okay, which is more suitable for each of the protein. Right. And when we have a certain protein is activated once it's folded, okay, this protein is actually they have their own family. Okay, protein ni dia ada family jugalah. Okay, macam jugalah lahir-lahir je ada family kan. So the same thing goes to the protein. So dia ada protein family. So if the protein functions in a certain way because of the sequence, because of the 3D structure that they have, so they are going to fall into a similar or the same category of protein family. So that is the protein. Okay. So meaning that the protein family in the same family, they actually share a common ancestor or same homology. Yeah? Because they have a similar 3D structures, function, and significant sequence similarity. So that's why they are going to be in the same protein family. And so, so why is that we can say that a certain protein is fall into a similar protein family or fall into the same protein family? Because the proteins, they are going to have almost highly similar protein sequence. So like this, for example, we have we have these mammals, different mammals, okay, different types of mammals, Homo sapien, there's so many different types here, but then we are under the same family, okay, because we have this similarity of the sequence, yeah. So it's just a small difference, just a slightly different, okay, slightly different in this 185 sequence, okay, amino acid sequence. This is just cuma sebahagian lah, okay, kalau nak tunjuk semua sequence dalam memos tu berjuta-juta sequence. It I just take a small portion of the sequence just to show apa ni organisms or mammals, ya, dengan contohnya manusia dengan mammals yang lain. Okay. And if let's say they have this kind of sequence, so perhaps kind of enzymes in humans, let's say, and we have this kind of substrate. And perhaps in another mammals, they are going to behave similarly like this. Okay, same thing when they behave in human or in the rats or in the zebrafish or in another mammals as well. So that's why we can for a certain drugs, that's why we test it on the mice, equals mama, and also now we have another another uh, another way how to diagnose the effects of the certain drugs to humans. Sebab kita boleh guna sekarang ni a new technology, we can even use the zebrafish. Okay, zebrafish. Right. Okay. So if a certain protein, they have higher similarity in terms of the sequence, and of course they are going to, we can actually see the relationship of the certain proteins by looking at the phylogenetic tree. Okay, we can find out the how similar the protein is from the phylogenetic tree, and perhaps the higher the similarity of the sequence, they're going to be in the same cluster like this. If let's say a certain proteins they have a highly different sequence, perhaps we're going to see that the protein is in this cluster and another one is going to perhaps over here. So by using phylogenetic tree, we can trace the ancestor of certain protein, where they are actually comes from, what are the sources of the protein, and so on. Okay, and this is the related to the highly sequence, high similar sequence of the proteins. So if let's say they have high similarity, so perhaps they are going to have almost similar structure, almost 3D structure. So if you can see over here, we have you can see the base, the base of the protein, the base part, which is they have the alpha helix, beta sheets over here. There's all this, they have the similar configurations, but perhaps the part that might be different is perhaps the coil. Okay, the coil part. Okay, the coil part. So meaning that perhaps they have they have a low conservation, okay, over the coil part. Okay, the parts yang berbeza tu, kemungkinan they are going to have a lower percentage of conservations, right? So enzymes. Okay, one of the protein is the enzymes lah. So this is a very common examples when we talk about the proteins. So proteins mainly are going to be the enzymes, because enzymes is works as a biocatalyst in so many pathways in the metabolisms. Okay, and I think you know about this, right? So all enzymes are actually proteins. Yeah, actually protein, but not all proteins are enzymes. Okay, so this is very basic. And enzymes are catalyzed. Okay, not all proteins are enzymes. So those proteins that not enzyme is not a biocatalyst. So those enzyme or those protein that can act as catalyst is actually an enzyme. Yeah. So we know that catalyst is a substance which increase the rate of chemical reaction. So that's why we can say dia adalah sebagai pemangkin, as a catalyst. So one of the properties or characteristic of the catalyzer, they are not going to use up. Okay? So at the end of the reactions, the the total number of the catalyzer is going to be just the same. It's not going to be used up. Okay. And okay, so when we talk about the proteins, I just want to explain on the method to visualize the protein. So actually, we have so many methods to visualize the proteins. Since okay, perhaps you are not sure why we can say that a certain protein have this kind of structure. Why? From how we determine it. Okay, how do we know that they act such they can be in this kind of configuration. So what type of method we actually have. So um so one of the method actually to visualize a protein, which is the most I can say this is the most advanced one, okay, most advanced method, which is the X-ray crystallography. So this is the method how to visualize a protein. So the first thing is that we need to have the protein crystal. So how to get the protein crystal? Okay, so you can have your target protein, your interest protein. To purify, you have to purify your target protein, and once it's purified, okay, 99% purified, for example, then you have to crystallize it. Okay, you have to mix with any of the kita kata adalah solution dia, and then let it in a certain temperature and it will become crystallized. Okay, so once the protein is crystallized, then we are going to do X-ray diffraction. So the same thing lah, macam you pergi buat X-ray, so you X-ray your body, then you can see what happen inside your body. Ada apa ni ada patah tulang ke mana-mana kan. So the same thing goes to protein crystal lah. So we are going to crystallize it. So the thing is that for the protein crystallization, for the protein diffraction, the wavelength is going to be different lah. Okay, the wavelength is going to be different. We need a very fine. We are going to need a source of the energy that can measure a very tiny measurement, yeah, a very tiny measurement. So you can imagine the distance between one amino acids to one amino acid is just 0. Perhaps 1.5 angstrom. So we are going to need such wavelength that can measure very tiny or very, sangat kecil measurement dia. Okay, so we are going to expose with X-ray. Okay, X-ray diffraction. Of course, this is different wavelength. Bukan wavelength yang kita guna untuk to X-ray your body or other types. So this is the memang memerlukan alat yang sangat advanced. Yeah. And we get the electron density map, then we come up with the computational sikit lah, computational work, molecular modeling, then we can get the final structure lah, final model of the structure of your target protein. Okay, so you know what, the X-ray diffraction, the machine that most advanced, we have in, I think the most powerful one is the one in Spring 8 in Japan. Okay, so the facilities itself, is built around a mountain. Okay, you can imagine it has a round shape of building, and the X-ray source is circulate in that building. So we need such kind of very big building, a very powerful source of X-ray to actually shoot the protein crystal, right? So it's very expensive, a super expensive experiment as well. So this is how we end up getting the molecule to the whatever protein structure lah. Okay, but then right now, not just X-ray crystallography. Somehow, right now we have also electron microscopy as well, also available for protein detection or protein determination. But that one is meant for larger larger molecules of protein, which is larger size, larger macromolecules. But for certain proteins that smaller, okay, which are small molecular size, still we need this kind of X-ray crystallography method. Yeah. Okay, so as for the conclusions, we know that proteins are built as a chain of amino acids, which then fall into a certain unique three-dimensional shapes, which is depends on the environment, depends on the side chains of the amino acids, which contain in the long polypeptides. Yeah, and we know that the bonding within the protein molecule itself will help the protein to be stabilized. And final for the form of the protein, of course, it will help to adapt to environments and will determine its function. Okay, so all right, so that's all for this topic lah. Okay, so we dah habis dah slides. Okay, any questions regarding the topic that we have discussed today? So ada 26 slides here. Yeah. Anything that you want to ask regarding the topics today? And the notes, I think these slides I already uploaded in the Putra Blaster. So you can get it from there, from the Putra Blast. So anything that you want to ask? Tak ada? Alright, if no questions, if no questions, tak sure lah. Just 46 of you. Tak sure siapa yang tak hadir ni. Okay, we don't have lab on Wednesday, so lab dah habis dah. But surely I am going to give you an assignment for that, as to replace the lab practical session. Alright. Okay, so if no question dah, enjoy your day. Okay, selamat Hari Raya. Jangan beraya banyak-banyak sangat.
Thank you, Dr.
Thank you, Doctor.
Thank you,
Alright.
Doctor.
Thank you, Doctor.
Thank you, Doctor.
Hmm.